Well, good afternoon. Thank you so much again for joining us. And we're looking forward to a very exciting and insightful talk from Dr. Francis Collins. But before Dr. Francis Collins comes up to talk, I want to introduce the next person. And he said, keep it short. So I'm going to keep it very short. It's my honor to introduce the person that this building is named after, Senator Roy Blunt. Senator Blunt.
Well, we'll just assume in terms of introduction, the building speaks for itself. How is that? What a thrilling thing for me. And again, I'm so grateful to all of you, not only that were involved in that decision, but in what you're creating here at this building. I'm not sure I can add a lot to the great job Dr. Altus did today introducing Dr. Collins. I'm going to try to do that. Before I do that, let me mention a couple of things that later I realized I should have done. There is a long list of things we've been able to do to help the University of Missouri system generally and Columbia specifically. And the real reason for that is my great staff, and particularly my staff here. Derek Coates, Sarah Graff have always been looking for the opportunity here and there, and I think there's seldom been a year when we weren't looking at a handful of different things for the University of Missouri at Columbia. And of course, as Dr. Collins and I were talking a little earlier in a meeting we're in, there are so many different things that happen here, and I think they really, as it relates to what we're talking about today, come together in an incredible way. You know, you've got a School of Veterinary Medicine on this campus, a medical school on this campus, you've got the reactor on this campus, you've got ag and food research on this campus, you've got extension on this campus, all of which come together around how we live and how we can live better. And I think there are very few places that bring all of those things together, and very few places, at least in the current environment, that encourage all those people to work together better than the University of Missouri does. President Choi has done an incredible job of bringing energy and vitality and commitment and a sense of the future to the university. And if you couldn't feel it here today, you just weren't paying attention of what is happening and how it can happen. Talking about Dr. Collins, and again, he's been well introduced today, and nobody speaks better for Francis Collins than Francis Collins, as we all saw for those minutes this morning. He's the only NIH director that served this long, 12 years for 3 different administrations. No other NIH director has ever served in 2 administrations, and Dr. Collins clearly could have served in much longer in this one. The level of respect, the strength of his personality, the things that come together to make him the leader that he is really extraordinary. The discussion this morning about the Shark Tank, I think creates a real pattern and model that the federal government can use as we move forward in President Biden's idea of a advanced research for health. You know, for half a century we've had advanced research for defense, DARPA, where the federal government didn't just give you a grant and check occasionally to see how you were doing, but became a real partner. And the Shark Tank, with 709 applications within the first 12 days or so, 150 of those made it all the way to the week-long Shark Tank, 50 or so made it to the month-long Shark Tank, and 31 of them produced 2 million tests every single day that we wouldn't have without that effort, with still the Shark Tank partners being active partners. And all of the home tests came from that. And it's a very hands-on kind of thing that Dr. Collins managed that shows what the federal government could do if they are given a challenge, or frankly, if they see a promise of something that can happen if we really make a full-fledged effort to make it happen. I hope we can maintain that pattern. Some people, not everybody, was disappointed when I said I wasn't going to run again. Many more people were disappointed when Francis said this would be his last year. But I think he helped establish something that can be very much part of the future, not part of what NIH does today, but something in addition to what NIH does today. And let me say before I introduce him how much I personally appreciate him being here. He and his wife are good friends of ours. They've been good friends of ours for 15 years or so. And he means a lot to me, but I think this is his first travel as director since March of 2019. You know, most of the federal government's still working somewhere else except where they used to work. And for this to be his first time on the road, for him to be here at this important time for the university, and I think an important time for the future of health in America. I think all of us, I hope we're all going to be able to look back and see that the curator's vision of what could happen, President Choi's vision of what can happen, really did set a new standard that I hope nobody else can beat. And the way nobody else can beat it is if we just keep doing better and better than we thought we could possibly do in this great facility with this great idea of a next generation of personalized medicine. Nobody understands that better. Nobody made that more possible by the Genome Project than the speaker we have here with us today. And when Abby and I leave in 10 or 15 minutes to get to St. Louis to get on an airplane, it's not out of disrespect or because we're bored. It's because we have to go. But Francis Collins, thank you so much for being here today.
That was incredibly generous. Senator, thank you for an incredibly generous and personal introduction. That means a lot. And I can't tell you how much your leadership and your friendship over these years has mattered and made consequences for the entire American biomedical research enterprise come true that otherwise simply wouldn't have. And I think what we're here today to celebrate this amazing building and all that it means for the science that's going to go on here is a wonderful concrete example of things that are happening all over because of your belief in the biomedical research enterprise and your willingness to make that a priority and not just something that would be nice to have. And you've brought a lot of other members of Congress along with you by your ability to articulate that. And it's pretty hard to resist the Roy Blunt pitch when he goes after you about why medical research is maybe one of the most important investments the government makes. Maybe even, I would dare to say, the most important investment when you talk about health, when you talk about life and death, and you talk about all the other things that come out of this in terms of American leadership and in terms of our economic opportunities as well. So thank you for that. I've learned a lot from you in terms of how best to negotiate through choppy waters, and we've had plenty of choppy waters, and we have quite a few right now. But having you as a partner has just meant the world, and being able to have this as also a friendship with my wife, Diane, and your wonderful wife, Abby, and to be able to brainstorm about things and put ourselves into really important missions like the Children's Inn has made it even better. So thank you. And I know you guys have to run off to a St. Louis plane to catch, but I do have at least one picture of you to show, so don't go quite yet. I'm really glad to be able to be here. And what a day it's been. I've really enjoyed the opportunities I've had to meet with some of your researchers, your center directors, get a brief dash through the building tour of this beautiful space that you have here, which any young scientist would just be salivating to see the way in which your laboratories are set up, with access to all kinds of other facilities. My gosh, your cryo-EM facility downstairs is like something that many people dream of, and you have it. There it's getting put together right now. And all of the other capabilities that you have because of the adjacencies on this campus, as was already enumerated by Senator Blunt, puts you in a unique position to lead. And this building should be an incredible magnet for talent. And so even as you bring some of your folks who are already here into space that I know they're really excited about being in, I know you're also going to use this as an opportunity to bring in a lot of other investigators of various sorts to fill out the capabilities that an institution like this can start to take on. And having heard from the president that funding from NIH went up by 88%. It's currently, by the way, $128 million this past year. I looked that up. I will predict that that slope is going to go even steeper upward with the abilities you're going to have in a facility like this to really build your remarkable contributions at a time where biomedical research has never been more exciting than right now. So it's the right time, and this is clearly going to be one of the best places to be to live out those dreams. And I'm glad I got a chance to come and see a little bit of it. So I did get asked to give you a little chat here about precision health. I will try to do that. I'll start out with a brief kind of comment on NIH in general. I think because a lot of people are interested, I'll give a bit of a rundown about what we have been doing about COVID-19 as an example of how this institution, when called upon, can bring science to the fore in a way that's really been quite remarkable. And then I will move into the precision health issue and a little bit of personal reflection on how we got to where we are and where we could go next with that. And maybe at the end, I'll even highlight a few things that your investigators are doing, because I thought it'd be nice to represent a little bit of the work that's going on here. So let's see how that goes. So yes, here we are in this beautiful building, and it is really quite remarkable, and glad to have a chance to help you celebrate it. But I also want to celebrate the person it's named for. And as you've heard, Senator Blunt and I have been working together in biomedical research issues for some time. You can see there on the left is a panel of quite a number of senators. I could do a quiz and see how many of you could name them. Gathered around a conference table at the NIH in one of our new research buildings. And this is one of the many visits that Senator Blunt organized to be sure his colleagues could come and see what we do there as well. And we would show all kinds of examples of what's going on in particular areas of research and usually have a great discussion as a result. But as I also mentioned, he's interested not just in science as an intellectual exercise, but what it can do for people. And I'm very fond of this picture. This is the Children's Inn at NIH, which is on the campus, but funded only by philanthropy. And this is where kids like the little boy you see there, who have basically no other medical options, come to NIH for participation in clinical trials, many of which we don't know if they're going to work or not. And they come with their families, and they stay at this Children's Inn. And during the day, they walk up the hill to the clinical center and go through all kinds of interventions. And the senator and his wife, Abby, have been wonderful supporters of this effort. And here's just one picture of the many visits they have made to this place. And Diane and I often go to the Children's Inn when we are needing to be reminded about why we're doing this all anyway, because it's, you just walk in the door and it becomes very clear. So NIH, as most of you know, has a dual mission. It's fundamental knowledge. That's our basic science effort. And we need to keep remembering that is the foundation of everything. The breakthroughs from 20 years from now will be because of the basic science that we're funding now and next year and the year after that. So that's about half of our budget. But the other half is the application, trying to move that swiftly in the direction of benefits for people to reduce illness and disability. And that ratio, sort of 50-50, has been pretty steady over the course of quite a number of years. As you know, NIH spends most of its money in grants out to places like this. Your $128 million is part of last year's budget of about $41 billion. And about 83% of our budget is of that sort. It goes out there to institutions who come to us with their best ideas, and we put them through the most rigorous peer review system in the world, and then we try to fund the ones that we think are the most meritorious. And it's always challenging because we'd love to fund about a third, or maybe even a little more than a third of the grants we get, because we know that's all good science. And gosh, 6 years ago, we were only funding about 15%, about 1 out of 6. Thanks to Senator Blunt and the leadership he had and others in the Congress who came alongside, we've improved that a lot so that we're now up in the low 20%. We're still not at 30. I keep hoping that's going to happen. Maybe what's coming in the next few years will get us there. We also have our own laboratories in the so-called intramural program. That's what you see in the picture. That brick building that you see is where my lab is. That's building 50 on the NIH campus. And that is a pretty amazing critical mass. And it also is the largest research hospital in the world, the clinical center at NIH, which has a lot of similar features to what you're building here, of having research patients who are right around the corner from research laboratories. So you can go back and forth, and it's fantastic for clinician researchers. We think also a part of our job is training. We do a lot of that, and communication. And I always have to crow about how our researchers seem to be doing pretty well by counting Nobel Prizes. And in fact, just 2 weeks ago, we had a trifecta where we had NIH grantees recognized for the Nobel Prize for Physiology and Medicine, for the Nobel Prize for Chemistry, and, wait for it, the Nobel Prize for Economics. Yes, that too. So we don't do that all that often. That was kind of nice. 5 more added to our list. So it's an amazing place with amazing talent, vision, promise for the future, all of that. And we had to bring that to bear on the worst pandemic in more than a century. And so I thought I'd walk you through fairly quickly some of the things that the scientific community did to try to respond to this. And I cannot say enough about how people rose to that occasion. Oh my gosh, people dropped everything, even those who hadn't previously done much in the way of working on infectious disease or the immune system or virology. And they learned quickly, and they figured out how their skills could be applied. And they didn't mind working 100 hours a week. Many of us have been doing that for the last 22 months. And look what happened. So this all started, of course, January 2020. These reports about something happening in Wuhan that sounded like it might be a serious outbreak. And at that point, all of us in leadership at NIH looking at these reports were beginning to think this might be the one that we've been worried about for years. We didn't really think it was going to be a coronavirus, although it could be. Everybody thought it would be influenza, and that might still be the next one. But we had reason to think coronavirus was on the list because SARS was a coronavirus and MERS was a coronavirus, and we had invested in coronavirus research over 25 years, and it's a darn good thing. Because when it became clear, when the sequence of this virus was published on January 10th, that it was a coronavirus, we knew exactly what to do. And within 24 hours, the vaccine was being designed using the mRNA strategy that had already been piloted but never applied in a human trial for SARS and MERS. So I want to tell you a little bit about what we did in these 3 legs of the stool here: diagnostics, therapeutics, and vaccines. I'll start with the vaccines and the therapeutics. And I have to say, a lot of this came about because of the willingness of everybody to collaborate in ways that were not at all traditional in the past. I'll tell you a little bit more about ACTIV in a minute, but this was an unprecedented public-private partnership between NIH, FDA, CDC, and about 20 pharmaceutical companies, again, sitting around the same table saying, what can we do together? Because we don't have time to waste. And then with diagnostics, as you heard this morning a little bit, this program called REDx, for which Senators Blunt and Alexander are in fact the main parents, we were able to plunge in with this remarkable Shark Tank approach, and I'll say a little bit more about the details of that also in a minute. But let's start with the vaccines. So the messenger RNA approach was in fact initiated almost immediately based on research that had been done at NIH, collaborating then with the company Moderna, and Pfizer picked up the same strategy almost immediately. But there are other vaccine strategies that were also being pursued, and one of the really wise decisions of Operation Warp Speed was to invest in all 6 of these, not knowing which one was going to work or whether any of them were going to work, because we had no reason to really be sure. And anybody who'd worked in vaccine development before would tell you that this is a trail that is littered with disasters and tears, because oftentimes things that you thought were going to be just right fail to deliver. So making sort of 6 shots on goal seemed like a good idea. The amazing thing is, of what you see here, virtually all of these have in one form or another shown great promise or have been approved. But the mRNA vaccines got there first. because the design was so quick, you didn't have to have the virus in your lab. You just had to know the letters of its genome sequence. And then you could synthesize that particular messenger RNA that codes for the spike protein, which is, of course, the thing that you want your immune system to have seen before and to react to and to be ready to react if it sees it again. And it's a very safe way of doing things because you're giving the ability through the mRNA for your own muscles to make that spike protein, but there's no way you can give somebody the disease that way. They're not getting anything except just the coding for that one protein. And again, years of basic science research making it possible. So when people say, well, gosh, did you just make this up on the night of January 10th, 2020? No. It was built on this. And there will be, I think, a Nobel Prize, maybe we thought it might be this year, maybe next year, for Katalin Karikó and Drew Weissman, who figured out how the mRNA approach could actually work in practice. And that was quite a few years ago. So this was then put into place. There's a lot of background here in terms of how this was developed to the point. There's the paper from Karikó and Weissman that showed how you could suppress what otherwise would be a reaction that would make mRNA not work. Structural biology was critical. There was a trick that Barney Graham at the NIH Vaccine Research Center developed so that you could basically modify the coding for that spike protein just right so that it would produce a protein in the right configuration for the immune system to recognize it. And that meant we needed to have structure. So yes, here we are, cryo-EM. Without this cryo-EM structure of the spike, I think we would not have had the successful design that we did, and there it was just in time. So out of that came the preliminary report, and of course, ultimately, in December, 11 months after starting this effort, FDA clearance for emergency use of both the Pfizer and the Moderna vaccine with efficacies that were breathtaking. Tony Fauci and I would sit around during this incredibly intense time when we're running the phase 3 trials with industry, trying to guess, okay, how's it going to turn out? What would be good enough to feel like, okay, this is going to be a way we could bring this pandemic to an end? And I would say, if we got 70%, I think I'd be feeling pretty good based on the way a lot of vaccines have not even gotten to that level. And Tony would say, oh, come on, let's go for 80. Well, look what we got, 94, 95%. Breathtakingly effective in that space. And so there we were in December with the opportunity for this to start going into people's arms, and it did. Now that was the scientific success story. Obviously, the way this is played out in the United States of America and across the world has been heartbreaking in every way. That's the graph for what it's been like in the US. You can see the terrible peak that happened there back last winter. You can see how things seem to be getting better, and maybe by June or July we were all feeling like, oh, we're out of the woods. And then Delta, that variant that's highly contagious, came along. And sadly at that point, we were not sufficiently vaccinated to prevent what happened, which is then this very steep upward peak, which happily now is coming down, but you can see it's still got a long way to go to get down to a point where you'd feel like, oh, we're really over this. You can see from the other graphs what's happened with hospitalizations and with deaths. Most heartbreaking of all, this is data from yesterday. 1,528 people died yesterday from COVID-19. 90% of them were unvaccinated, and that's the case all the way along here in terms of the people we are losing. Now you can see 724,499 people have died from COVID-19, more than 100,000 of them unnecessarily because of deciding not to take advantage of the vaccine. That's the part I didn't really anticipate. I thought if science rose to the occasion, we spent all of our time trying to make sure that it would be so compelling that everybody would embrace this opportunity to save themselves, their families, their communities, especially thinking about the people around them who are more vulnerable. People like Colin Powell, who we lost yesterday, who had multiple myeloma and was one of those vulnerable people. And I don't know how he got exposed, but it's a terrible loss that this has happened. And somehow we have failed to be able to communicate effectively to a large segment of the population, about 65 million people in the United States who have yet to decide to roll up their sleeves. And there's a whole lot of complicated reasons for that relate to trust, or more likely distrust, to information that gets spread rapidly through social media that is frankly false, and some of which is, I think, intentionally being propagated by people who know it's false. And those are the people I really have a hard time forgiving. I recognize that an awful lot of people are simply barraged by information that causes them to be fearful and confused and a bit paralyzed. And I'm totally sympathetic there. I just wish somehow we had anticipated this in a way that would have given us a better chance to win the case for what the facts are, because the facts have oftentimes been pushed aside by various opinions, conspiracies, et cetera. So this is a sad part of this. Are we making any progress? Well, vaccine doses, I go back to April, I was feeling pretty good. Look at this. We were doing like 4 million doses a day. If we'd stayed on that curve a little longer, this would be a very different world right now. But we kind of hit the wall. And at that point, the people who were interested had kind of already done their thing. And then the numbers start to drop back, and you can see, even now, when the information out there you would think is so compelling. I mean, if you see 1,500 people a day dying and they're virtually all unvaccinated, how would that not motivate you to decide to take your own steps? But we haven't gotten there. And of course, now we have mandates. I wish we didn't. I wish we didn't have to. But if that's what it takes to somehow get past this and to save those who are vulnerable and can't be protected, either because they have immune deficiencies or they're too young, then isn't that also kind of a community responsibility? And it certainly has been used in other ways for other diseases, as we are now trying to do for this one. I guess you can hear a little bit of my frustration here. This is not the scenario I thought I'd be talking about in October of 2021. But let's go back to other aspects of this. I want to tell you a bit about therapeutics, because it's fine to say we're going to prevent the disease, and believe me, we will push that as far as we can, but people are still going to be getting COVID-19, especially in other parts of the world that have yet to have much access to vaccines at all. And what are we offering them? So what to do about that? This is where this active public-private partnership came in. I looked around with my colleagues in the spring, early spring of 2020, and the efforts to try to find treatments for COVID-19 frankly, were a scattershot. There are a lot of well-intentioned efforts picking at something that might work. Many of those were small studies that probably were not well enough powered to give you a compelling answer. Many of them were focused on exactly one compound, hydroxychloroquine, because people thought maybe that's it. And we now know that was not it. And so we had to come up with some kind of strategy here that would bring the capacity of clinical research in a coordinated way to see what might actually work. So I pulled this group together of public-private partners. This now involves, and almost immediately did, 20 industry leaders, and these are basically the chief scientific officers of this partnership who have put their time and effort into this. So it's very much endorsed and supported by all those companies and all the government agencies you see here and nonprofits. And the foundation for NIH, which is a nonprofit that has exquisite program management skills, took on that role. And this group met every day for a number of weeks, developed 4 different working groups that are still meeting several times a week. This is probably 100, 150 people, most of them fairly high-level scientists in these various organizations who have put everything else aside to drive this engine called ACTIVE. So what do we need to do? First of all, we had to say, what are the most promising therapeutic agents anyway? Well, made a list. There were about 620 that people had in one way or another said, well, that might work. These are mostly what you'd call repurposing. So compounds that have been used for some other human illness that maybe might turn out to be effective against COVID-19, but you don't know until you run the trial. So, okay, let's prioritize that. And that meant going through more than 600 files of possible evidence and deciding which were the most promising. But then you had to run the trials. So that meant pulling a very complicated clinical trial network across the country supported by NIH and industry, and turning it into something that could move on a dime and be basically starting a trial within a couple of weeks for a new idea for a therapeutic that had just emerged at the top of the list. And you had to have master protocols so that there was going to be no question about how the trial was run, how rigorous it had to be. And FDA was right at the table for that, helping us design those master protocols so that if the trial was won that way, FDA would say, okay, we'll accept the result. You don't want to make the mistake of thinking you had the design right and then FDA telling you, no, start over. That was an amazing experience. And within about a month after ACTIV got started, the first trials were getting underway. This is still in very much active space right now. We are running a total of 6 master protocols right now today. And what we've learned about it, I would say we have some singles and doubles. We don't have home runs. Monoclonal antibodies were the best we got. And we got some pretty good stuff out of that. But you know what we learned about monoclonal antibodies? They don't work if you wait past the first week, which means they don't work at much of all in hospitalized patients. And why is that? It's because your own immune system by then has already generated antibodies. And by adding additional ones from a pharmaceutical product, it doesn't seem to help in terms of response. Where you get benefit from monoclonal antibodies is give them early in the first 5 days of illness, especially to high-risk people, and then you can save lives. But it took a lot of trials to find that out. I guess if I was going to try to summarize those more than 20 therapeutics, what we've learned is that it depends on where you are in the clinical course, which therapy is going to work. And all of this is now much more documented in the clinical trial, in the clinical guidelines that are available on the NIH website. So if you look from left to right here, starting with somebody who's asymptomatic but infected, all the way to somebody who's on a ventilator, the early stage is best for the antiviral strategies, like monoclonal antibodies, like the drug I'm coming to in a minute, Malnuvir. But if you wait until somebody is much sicker, the antiviral strategies may not work. By that time, the viral load in the person has actually gotten pretty well knocked back by the immune system. And it's the immune system itself that is causing the greatest havoc. And that is why you need then these immunomodulatory strategies like dexamethasone, a steroid, in order to stop the cytokine storm that is doing more harm than good. We also learned along the way, though, that this virus is very good at causing blood clotting, both in large vessels, but especially in small vessels, and that if you want to help people who are really pretty sick with this, anticoagulants could turn out to be beneficial. And yes, in rigorous trials, we have shown that is the case. If you just got admitted to the hospital with COVID-19, you're not yet in the ICU, you want to be on full dose heparin. That is going to improve your outcome. And we didn't know that until we ran the trial. Now, again, I said we got singles and doubles. We didn't get home runs. What we all hoped for was a small molecule oral antiviral that could be given immediately after a positive test and would basically drive the virus away 100% of the time. And none of our repurposed compounds did that. But we're determined to keep going on that road. And so there is now vigorously underway this APP antiviral program for pandemics, starting with SARS-CoV-2, but expecting this might also apply to other future pandemics. And we have to think about that right now. So this effort aims to try, again, working with industry, to identify those really much more specific targeted drugs that would interfere with the viral life cycle. And there's some very appealing targets in the life of SARS-CoV-2, particularly a couple of proteases, which if you could block those with a small molecule, you should stop that virus in its tracks, and you should not really have host consequences because it's the viral protein that you're hitting. It should not, therefore, have host side effects if you're really specific. And we're in the middle of doing that. And the first one to mention, which is now in the news a bit, is Malnuvir. And this, I'm proud to say, is a drug that was originally developed at Emory in their Drug Innovation Ventures group before COVID-19 came along, but it was intended to be an antiviral probably for flu. But then when you looked at its features, it looked as if this might potentially work against any RNA virus, because its mechanism is basically to interfere with RNA replication by inserting gibberish into the genome as it's trying to be copied. So it was tempting to think this might be useful, and here we are now, October. This eventually was licensed out to Merck. Merck ran this trial, which just was announced a couple of weeks ago, with pretty interesting results. 1,500 patients, half of them got Malnuvir, half got a placebo, and when they looked at the data, this reduced hospitalization by 50%, which is a pretty big effect size. Again, the time to do this is right after diagnosis. This is an antiviral. We know you got to give those early. That's what this trial did. So they've sent their data to FDA, and FDA is reviewing it, and we will see whether FDA thinks that's good enough to grant emergency use. That'll be a game changer. That'll be our first oral agent for people just diagnosed. And I'm a little worried here that then the people who haven't gotten vaccinated will say, well, there's one more reason I don't have to worry because now we have a pill. No, wrong answer. You don't want to get this disease. And this is 50% reduction of hospitalization, but the other 50% go on and get really sick, and some of them die. So yeah, we need this, but if you have to make a decision here, get vaccinated. Other drugs are coming along. Pfizer and Roche both have now antivirals that are looking promising. There'll be more to say about those, and a lot more right behind that we are deeply engaged in to try to be sure we have a real toolbox here to help as this disease continues, because it's going to continue at the rate we're going. Well, let me say something about RADx. And again, this is the brainchild of Roy Blunt and Lamar Alexander, who in a conversation with me back in April of 2020 made it clear that they thought something more ought to be done to develop diagnostics beyond what we had at that point, and NIH ought to consider coming up with a new strategy for that. And out of that was born this idea of the Shark Tank. And again, this RADx initiative aims to try to accelerate innovation in this space. And we knew there were people working on this, but most of them in academic labs or small businesses didn't really have a path forward to get validated and then to be able to expand. So that's what the Shark Tank idea was supposed to fill, and it's a Shark Tank which is also an innovation funnel, if you'll forgive me for mixing metaphors here. And there's a lot on this slide, but again, this was an idea of inviting anybody who's got a great idea about technologies for detecting SARS-CoV-2, bring them forward. Ultimately, we got 3,000 people starting applications, 824 complete applications. 179 of those went into the Shark Tank. It's interesting hearing Senator Blunt reel off these numbers like he has those all in his head, which is pretty impressive. And in the Shark Tank, 47 of them came out saying, yeah, you got something here. And at that point, we helped them get FDA validation. We gave them money to help them scale up. We helped them deal with things like supply chain and manufacturing. And those now 35 of those are now fully approved and out in the clinic, giving rise to this 2 million tests a day that you heard about, which has been just an amazing experience. So that was a quick romp through where we are, diagnostics, therapeutics, vaccines for COVID. But of course, many of us are really thinking hard now about the lessons learned, because even though we're still in this one, there's going to be another pandemic, and that we cannot make the mistake this time of slipping back into complacency. I don't know whether it's 5 years from now or 10 years from now or 2 months from now, but something else is going to emerge. Maybe the next one will be a coronavirus, maybe it'll be flu, maybe it'll be a filovirus. You could make a list of the 20 most likely viral families, and you probably have a pretty good list. So should we start now to develop antivirals? Should we start now to build vaccines? I think we should. And that's going to take resources. But if we did that, and then one of these emerges, we'd be months ahead of where we otherwise would be. And I guess I've learned with COVID-19, every day matters with lives at stake. So I think this is something we should do, and the government, most part, seems to agree. We will see whether some of this gets funded in that reconciliation package that you keep hearing about. I hope it doesn't get lost along the way. Okay, well, that was a not terribly brief detour into COVID-19, but I thought you all might be interested to hear how that has played out, because it is connected to precision medicine, indeed, trying to figure out in the most precise way how you could intervene with a terrible pandemic. But let me say something more directly now about precision medicine, precision health. Again, the vision, the traditional medicine view was mostly the expected response of the average patient. That's mostly what we do in medicine because that's the evidence we've got. But you want to move beyond that to these individual differences in genes, environment, and lifestyle to enable more precise ways of not just treating but preventing disease. Because I ran the Genome Project, people think, oh, it's just all about genomics. Well, no, that's in there. That's a factor, but that's far from the whole story. This is about environment. This is about lifestyle. This is about health behaviors. This is about social determinants of health. This is about health disparities. It's about all of that if we're going to be serious about precision health. But how do we get there? Well, one way for the genomics part we got there was the Genome Project. And I did have the privilege for an amazing 10 years of overseeing this from 93 to 2003 and to be able to bring together ultimately 2,400 scientists in 6 countries to do something that seemed impossible at the beginning, read out those 3 billion letters of the human genome. Again, that cost us about $3 billion over the course of those 13 years. You can now have your human genome sequence read out for about $500, and you can get it in about 2 days. I don't know of any other technology that has ever moved that quickly in terms of reduction of cost and speed of delivery, and at the same time, the accuracy is much better. So people say you can't get faster, better, and cheaper. We got all 3. And it's getting better all the time. So the Genome Project was this amazing team effort, and I learned a lot about teams from this particular enterprise and the ability then to deliver something to the public. And the other thing we did, which has become, I think, kind of a standard now for a lot of these large-scale, big science biology projects, is we gave all the data away. That was critical. We put the sequence up on the internet every 24 hours so that anybody that might start using it could get access to it and not waiting for publications that might have taken much longer. It's been 20 years. So yes, the first publication of this in Nature, February 2001, and there's an awful lot that's happened since then. And it feeds directly into this idea of precision health, and it becomes so much more straightforward now to be able to derive genome sequence information going from left to right. That left is a photograph of the sequencing center at the Broad Institute, which generate a lot of the data, which is like a factory, and now it's something you hold in your hand. It's like just amazing. But I'm a physician. I wanted to see this not just as a triumph on an intellectual basis, but how is it going to help medicine? How are we going to give the doctors here at the foot of the bed a chance to understand what's actually being read out there in all those G's, T's, A's, and C's? So the other thing that needed to happen, and is still happening in a great many ways, and which this center, I think, this building can be part of for whatever the next generation is all these other projects that are going to decorate the genome with information about how it works. The variations in the genome that mean we're not quite the same, although we're darn close. The ways in which the genome has its function determined, not just by the genes that code for protein, but all that regulatory information, how it plays out in cancer and other diseases. So all of those things also now freely accessible, big projects that NIH mounted on the heels of the Human Genome Project. And now it seems like the time has come to try to pull all of that together to make this precision medicine, precision health dream come true. And it is the right time to do that because I think we are at a place where patients are increasingly interested in this. The access to electronic health records, which 20 years ago, very few people had one. Now most do, although they're still very clunky, and we have to think carefully about privacy in any kind of research application. All kinds of other technologies that allow you to collect data. I mean, my Apple Watch right here, if I wanted to, could give me an EKG right now. If I thought this talk was giving me an arrhythmia, I could quickly check and see if it was. And of course, genomics, and then all the ways in which data science and the ability to deal with very large data sets has made a difference. So one of the dreams I've had is how do you bring all that together in a large-scale project that would have open access to qualified researchers to really figure out what is it that allows people to stay healthy and causes others to fall ill. And that's where this project came out, called All of Us. And Missouri's had a role in this, so has whole the country. The idea here is, let's invite 1 million people to be our partners. And let's invite them to share everything about their health status, their electronic health records, filling out all kinds of questionnaires about their health behaviors. Let's get a blood sample and sequence their complete genomes. And let's get them okay with the idea that after we anonymize all their data, other researchers are going to look at it and try to learn what's going on. And with 1 million people, you're going to have incredible abilities to track over time what kind of factors are associated with health or with illness. It's historic, to put it mildly. We are aiming for 1 million. You can see where we are as of today, 415,000, so not quite halfway there. Some of them are not yet complete because they don't have yet provided a blood sample. We got slowed down a bit by COVID because people didn't want to go to a clinic for that sort of thing. But a lot of enthusiasm, a lot of participation, and already the researcher Workbench is open for people to work on this. The genomic information will be landing in this database in the next 2 or 3 months, which will get people even more interested in having a look at it. And again, the mission is simple: accelerate health research and medical breakthroughs, enabling individualized prevention, treatment, and care for all of us. Now, this will be a foundation, I would say, for all kinds of other studies. We're not going to be able, for instance, to do PET scans on 1 million people, but we might be able to invite 10,000 of them to do that. And all of these participants are pre-consented for recontact. In fact, they've said they're interested. So if there's another study you want to mount on a group of people on which you already have remarkably deep legacy data, here's the group you can approach. If you want to do a study on diabetes, well, you know, with 1 million people, there will be about 90,000 of them who have type 2 diabetes. That's a pretty good start for a clinical trial of some sort. If you want to try out some sort of new therapeutic or diagnostic, suppose you have what you think is a really good way to do ambulatory blood pressure measurement that really works. Well, these folks are really quite used to being asked about that sort of thing. They all are turning in their Fitbit data. We can simply add that on and see what the data looks like. So this is still in its sort of first 3 or 4 years of getting started, but this is going to be an engine for all sorts of things that I think here in this next-gen precision health program, you're going to want to pay close attention to, and which you will have close access to. Again, the Workbench is open. And Missouri is part of this. The kickoff of this on May 6th, 2018, included Union Station in Kansas City, and Mizzou has a data use and registration agreement with 2 of your researchers who are already currently using the data. WashU is also an important partner. This is intended to be geographically dispersed over the whole country, as it should be. And the other thing about this, there's a bunch of data here about what we ask people to contribute, which is probably more detail than you need, but it's a lot of information from each individual. And we have focused intensively on diversity as part of this. And if you can see the bars there, you'll see that less than 50% of these are individuals who are European white. The rest are of other race and ethnicity. And we have worked very hard to achieve this. And also by recruiting through things like the federally qualified health centers, have diversity in socioeconomic status and other kinds of social experiences and health disparities. So this is going to be, I think, a wonderful resource for trying to understand health inequities as well. Well, I wanted to walk you through a little bit of that, but I think the next generation of precision health is going to happen in places like this. I don't even know what you all are going to come up with. The people that you have here already probably have some ideas. Others that haven't arrived yet will bring other ideas, but it's all going to fit nicely in this theme. And I just thought I'd finish with a quick shout out to 3 investigators that I just read about who are here, who are doing things in this space, just to make it clear that I know this is not unfamiliar territory to you, but you're going to be expanding it. So this set of investigators shown here studying diabetes-related cardiovascular disease. What is going on there in terms of how diabetes triggers disruptions to the cardiovascular system, and how could that be intervened with to try to translate into better outcomes? And every diabetic is not the same as every other diabetic. So there you go. That's a precision health strategy. And again, these are among the first wave of researchers, I gather, who will move into this building. I hope I'm right about that. I hope they knew that already before just now. I have learned today just how much of a resource you've got here in terms of the research reactor and the way in which that provides opportunities for lots of applications, but maybe particularly for targeted cancer therapy. And again, that's going to require a careful analysis of which therapy is most appropriate for which patient. Okay, that's precision medicine. This will be a critical part of what you're able to do here that most other places are not going to have the capacity to do because they don't have a reactor like you do. And chemotherapy is obviously something we use, but we would love not to have to. And so I wanted to highlight this, which is currently, again, tapping into your close relationship with the vet school, how you might actually create a vaccine from the tumor to actually lead to cancer cells being targeted by immunotherapy, something that Roy Blunt mentioned this morning in his comments about how critically exciting cancer immunotherapy has become. And I just got to say, that mRNA approach to making vaccines, bring that to the cancer applications, and suddenly you can go a lot faster. Some of us are even contemplating a project that might go into this new part of NIH called ARPA-H that Senator Blunt was talking about. Why don't we try making a messenger RNA vaccine that basically would immunize you against the 50 most common mutations that happen in solid tumors? We know what they are. Just see what that would do. If you had that immunity, when that first cell popped up that had a RAS mutation, bam, got it. Would that actually turn out to be a phenomenal way to do prevention? I don't know. But the thing about mRNA vaccines is you can do it fast, and you can multiplex them. You don't have to decide, well, I'm just going to go for one target. I could do 50 at once just by mixing the mRNAs. Think about that. You'd want to start with animals first. I'm glad that that's why a place like this is all set up to address that. So that was kind of the end of where I wanted to go. I did want, though, to finish just by saying something about what we're all about here. And I'm very fond of this particular quote from the poet Peter Levi about hope. You know, National Institutes of Health, sometimes I like to think about is the National Institutes of Hope, and this button that I wear on my lapel next to the button I got from you all today it's a guitar pick. You might have heard that I'm also a musician, so it had to be something recognizable. It's a guitar pick, but what does it say? It says hope at NIH, because I think of what we are all doing is about hope, and NIH is the National Institutes of Hope if we're doing our job well. But people use the word hope sometimes in sort of careless ways. Well, I hope for this, I hope for that. No, Peter Levi says, hope in every sphere of life is a privilege that attaches to action. No action, no hope. Okay, those are strong words. But I know all of you at this institution and who will work in this incredible Roy Blunt Next Gen Precision Health building have hopes that what you are doing will actually save lives, will prevent suffering. Otherwise, I don't think you'd be doing what you're doing, because let's all recognize this is not easy stuff. It's hard doing biomedical research. Most of your experiments fail. Getting the funding that you need is not trivial. I'm sorry, I wish we were able to do better. Thank you, Congress, that we're doing better than we used to. This is a real struggle, and you've got to believe in it, and you've got to have hope that this is going to change things. Well, okay, your version of hope resonates with this particular motto. You are all about action. And the action that's going to happen here at Mizzou in this remarkable facility with all the collaborations around it, I think will be a reason for that hope to become a reality, because it's attached to that kind of action. So thank you for the chance to be able to spend this day with you, a remarkable day. And sometime I hope to come back and see what's happening as this building is filled up with people with amazing ideas, interesting experiments, new ideas of things they might want to do next, and hope that is turned into reality. I am sure that's what's going to happen here. Thank you all very much.
I do have a microphone if anybody has any questions.
Yes.
Thank you for that. This idea of studying a lot of people to understand individuals seems like a little bit of a paradox. Can you talk a little bit about that?
I totally hear you. And some people have even said, well, this is just not consistent with public health. You're undermining the whole idea. I think actually it's a paradox that we can build upon. So if you want to understand how individual differences play a role in health outcomes, you have to study different people to see how those correlations occur. And if those individual differences are complex, it's not like a binary, well, either you're a 0 or a 1, but it's a whole array of different data types, you're never going to sort it out unless you have a very large data set to work with. Then you can start to see correlations that otherwise would escape you. We've learned that, of course, in this whole effort to try to identify genetic factors involved in common disease, where when people started out looking for those in diseases like schizophrenia with maybe 100 people, came up empty, and then you got to 10,000 people, and oh my gosh, it all starts to happen. Same will be true with other kinds of factors involved in common diseases, whether they're environmental or health behaviors. You got to have the sort of power that comes from numbers before you can start to see the individual effect. But you will get to the individual effect, just like I did finding out my diabetes risk and avoiding the honey buns. That is ultimately what the goal is. But you don't get to the goal without starting with a really large collection of individuals, and then you can sort it out. But your point is very well taken, because it does seem like, wait a minute, what? It all fits.
Well, Dr. Collins, I just want to thank you so much on behalf of Dr. Choi and the University of Missouri for coming here today. We all have been anticipating this day as a very special day in the history of the University of Missouri, but having you here has just sort of been the honey bun on the cakes. And also, I think we all want to just express our thanks to you for all you've done for this country and for biomedical research throughout your career, for the NIH, but particularly at the NIH, for everything that it's meant to the lives of Americans and everyone around the world. So thank you very much, and good luck in January. So thank you
so much.